Hello, my name is Aisling Murren, and this presentation is about my ongoing work in the Fukami lab about the interactions of microbial species found in tree holes. Complicated communities are everywhere, even in the places we don't expect, like the water which collects in the crevices of oak trees. These tree holes are thriving with protists, whose interactions are complicated, nonlinear, and challenging to predict. Our work seeks to utilize this tiny, bustling community as an experimental microcosm to explore generalizable ecological principles. We were led by one main question in our research: how can we explore the impact of varying state-dependent interactions on community stability using tree holes as a model system? To shed light on this question, we gathered dry litter from oak trees in Jasper Ridge, rehydrated the detritus, and created tiny microcosms, ecosystems in a jar. Then, every day, at the same time of day, we sampled the communities, identified the protist species via microscopy, and counted the species abundance for each replica. Then, convergent cross-mapping, CCM, and SMAP, a nonlinear local weighted linear regression, was performed on this data to identify causality and interaction type linked species. CCM allowed us to build interaction webs based on the causality rather than just correlation. SMAP further qualifies the sign and strength of these interactions. Seen here, the web depicts the average strength of unidirectional interactions between species. As you can see by the weighting of the lines, the Cyclidium population really impacts the Heteronoma population. The weakest interactions were excluded from this web. That graph gave a rough idea of what's going on, but the fun begins when we look closer at the variation in interaction strength over time. As these graphs shows, interactions between species are not constant, even though we often treat them as such. They peak and drop and shift dynamically according to system states. This particular graph, we can see that Cyclidium and Spirostomum populations are strongly negatively related, but at other times they have almost no effect on each other. One biological explanation for these shifts is that the populations compete heavily for food when it's scarce, but neither population affects the other when there's plenty of food to go around. This nonlinear time series modeling research gives us better insight into the varying mechanisms driving population change. This has powerful applications in a predictive capacity. For example, mosquitoes rely on our study community as an important source of food. So moving forward, we plan to model the interactions between protists, bacteria, and mosquitoes to better predict mosquito population dynamics. More broadly, our microbiome research can be viewed as a case study for other larger ecosystems, which are more challenging to model because of scale limitations. Before I close, I would like to acknowledge that this research and my personal learning experience would not have been possible without support. Thank you so much to the Rose Hill Foundation, to my lab mates, and to the Stanford Biology Department. Thank you so much for listening to my presentation.